Namaste and welcome back to Typical Monk. Last class, we met the cell, the unit of life. Today we go inside the simplest cell, the prokaryotic cell, the blueprint of bacteria. It looks simple, but NEET loves to make questions from it. No hurry, one idea at a time. Here is today's path, 5 easy stops. First, the shapes and the small size of bacteria. Second, the cell envelope, that is the 3 outer layers. Third, the mesosome and the plasma membrane. Fourth, ribosomes, inclusion bodies and the nucleoid. And fifth, flagella, pili and fimbriae. Bacteria are the most abundant organisms on Earth, but they are easy to recognize because they have just 4 basic shapes. They are very small, only 3 to 5 micrometers. Remember 4 shapes: coccus, that is round; bacillus, that is rod-shaped; vibrio, that is comma-shaped; and spirillum, that is spiral. Simple in structure, but complex in behavior. They survive almost everywhere. The whole inner plan stays the same, only the outer form changes. Now let's move from outside to inside. The outer part is called the cell envelope. It has 3 layers. Outermost, the glycocalyx. It is either a loose slime layer or a tough capsule. Beneath it, the cell wall. It gives the cell its shape and stops it from bursting. And innermost, the plasma membrane, the true living boundary that decides what comes in and what goes out. Now, let's look at the cell wall a little more closely, because a very commonly asked question comes from here. The wall is made of peptidoglycan, a mesh-like material that gives bacteria their strength. In gram-positive bacteria, this wall is thick. They hold the stain and look violet. In gram-negative, the wall is thin, but has an extra outer membrane on top. They look pink. This stain test was made by a scientist named Graham, and he decides which medicine will work. Now, a special part of the membrane, the mesosome. The mesosome is really an inward fold of the plasma membrane, like a pocket pushed inward. It does many jobs in respiration, in forming the cell wall, and in copying the DNA during cell division. In some blue-green algae, folded membranes carry pigments. These are called chromatophores. Now inside the cell, where protein is made and where food is stored. The ribosomes of bacteria are the 70S type, made of a 50S and a 30S part. This is where protein is made. When many ribosomes sit on one message, it is called a polysome. Together they make protein. Inclusion bodies store food, and note they have no membrane around them. Examples: glycogen, phosphate granules, cyanophycean granules, and gas vacuoles. Now, the most important part, the genetic material and the thread sticking out. The DNA of bacteria is a single circular thread with no nuclear membrane. It is called the nucleoid. Some bacteria also have small extra rings of DNA called plasmids, which are very useful in biotechnology. Flagella, long threads that help the cell to move. And pili and fimbriae, small bristle-like structures meant for attachment, not for movement. Now, the high-yield pointers. The facts that NEET turns into questions. 1. The prokaryotic ribosome is 70S, that is 50S and 30S, while the eukaryotic one is 80S, that is the biggest trap. 2. The mesosome is a folded membrane that helps in respiration, wall formation, and DNA replication. 3. Inclusion bodies and the nucleoid both have no membrane. 4. Pili and fimbriae are for attachment, flagella are for movement. Do not mix them up. 5. The cell wall is made of peptidoglycan. Gram-positive is violet, gram-negative is pink. Now let's clear the mistakes students fall for. Myth: prokaryotes have no ribosomes at all. Truth: they have 70S ribosomes. They just lack membrane-bound organelle. Myth: bacteria swim using pili. Truth: pili and fimbriae are for attachment. The moving is done by flagella. Now, test yourself. Think of each answer before it appears. Answer first. That is how you build strong NEET recall. Question 1. What type is the prokaryotic ribosome? Answer: 70S. Question 2. What helps the DNA replication in bacteria? The mesosome. Question 3. What is the bacterial cell wall made of? Peptidoglycan. Question 4. What is for attachment, not movement? Pili and fimbriae. Question 5. The small extra ring of DNA in bacteria? The plasmid. Let's gather it all in one breath. 4 shapes: carcass, bacillus, vibrio, spirillum. 3 envelope layers: glycocalyx, cell wall, plasma membrane. The mesosome, a folded membrane doing many jobs. 70S ribosomes and inclusion bodies, and the nucleoid have no membrane. Flagella move, pili and fimbriae attach. In the next class, we go inside the eukaryotic cell, the membrane and the endomembrane system. See you there. You have understood the full blueprint of bacteria, the simplest cell, now fully clear. Be proud of that. One cell at a time with a calm mind, and you will surely crack NEET. This is Typical Monk. Every class, always free.